Hi, I'm Dr. Joe Lamb. Good to see you here today. This is our Lunch and Learn series coming to you from the Personalized Lifestyle Medicine Center by Metagenics in Gig Harbor, Washington. So today is our second in this series. The first one took place a month ago. On Thursdays, we do this Thursdays, third Thursday of the month at 12 noon. And the first talk a month ago was on what is functional medicine and why you need it. And today's talk is entitled, being mean computer. Today's talk is entitled An Introduction to Your Genes, Epigenetics, and Nutrition. And we're specifically focusing today on the connections between what our genes tell us about who we are and how they impact our lifestyle choices, and how they affect how we're going to manage our health in the future. So genetics has clearly changed in the last several hundred years. You know, we had Mendel writing in the 1600s, I believe, 1700s, about pea experiments. And maybe Mendel was even in the 1800s. I'll have to check that. But he did his experiments with peas and was trying to determine if he could select out different aspects through breeding. And he found that to be the case. And Darwin clearly noticed the variation between the species and came up with his natural selection law. And between the two of those, we started realizing what those mechanisms were for adaptability and change over time. And the consequences of Mendelian genetics and Darwinian kind of philosophy was that by the time I got to college, we pretty much believed that there was a determinism, a genetic determinism, that at the time of our birth, genes came from our parents, one set from dad, one set from mom. They merged, and as a consequence of that merger, a unique person was made, and a lot of their medical history was already written. A lot of that history was already out there and defined, and you were going to go down a certain path, almost like you turn on a computer and its operating system boots up, and it boots up the same way every time, and it's going to do certain tasks, and all you had to do was turn the on switch. And that moment of fertilization was that moment of the on switch being turned on. And that was really the thinking through the 1980s and into the early 1990s. And what defined the 1990s as being unique was the Human Genome Project. You know, we finally had the technology due to something called polymerase chain reaction, which I believe was a consequence of some work by a guy named Muller, something like that, sorry. But he was a scientist who came up with a technique which allowed us to replicate small amounts of DNA that you could find in a sample and get enough so that you could actually pull it apart and figure out what its structure was. And there were other techniques about cutting transcription pieces and taking little blocks that allowed us to decode DNA. And both the government-run program and the program run by Craig Ventnor went into competition in the 1990s to come up with the human genome. Now, the interesting thing was that there were a lot of surprises that came out of the Human Genome Project. And clearly, one of them was where variability comes from. It was recognized for a long time that there is a lot of uniqueness between human beings. And, as I frequently say during these talks when I'm talking to clinicians, if I'm teaching in a room and I've got about 50 physicians in a room and they're sitting at tables, by the end of the day, I can pretty much identify everybody in the room and where they're going to sit. So like when someone's coming back in a little late from lunch, I know who they are, I know what seat they're going to fill up, and I know who I'm still missing. Now, if I was teaching a room full of 50 fruit flies, like I had 50 of them right here in front of me on the table, I would not be able to identify them nearly as well. I wouldn't be able to see them as being unique. There wouldn't be enough variation to be able to label one Charlie, one George, one Mary, one Heather. I wouldn't be able to do that. Instead, they'd all look alike. So when the Human Genome Project started, the belief was that, in our DNA, we had these large sections of what we called at that point junk DNA. And then we had the sections that was the DNA that specifically coded for certain proteins or certain enzymes, and the proteins being the structural building blocks and enzymes being those tools that convert one piece of structure to another, or one piece of signaling molecule to another, or intermediates in a step, like as we're going from making cholesterol making vitamin D into cholesterol, and cholesterol eventually down the road into the steroidal hormones, the stress response hormones and the sex hormones, for example. And I said that wrong. Cholesterol is turned into vitamin D, not the other way around. So excuse me. Clearly, we thought the variation was coming about from the number of genes. And it was actually expected at one point in time, before in the late 80s, early 90s, that there would be maybe 600,000 to 700,000 genes that would code for all of the different proteins and the types of proteins that led to all the variation. So you can tell us all apart. By the time the project started, the thought was that there would probably be 100,000, which would define who and what we were compared to, other organisms. By the time the project was done, we were really surprised because we had about 30,000 genes, which puts us in the ballpark of as many genes as a fruit fly or a carrot actually has. Even a worm. We're not that many more proteins there that are, or enzymes that are creating our uniqueness. Instead, what was found was that there's a large degree of regulation in response to the environment, signaling molecules which sense what's out here and around us and allow the environment to have its effect upon what our genotype is. So genotype refers to what your genes call for, and phenotype is the consequence of the interaction of your genes with the environment. And so what ends up getting expressed is the organism. But it was fairly clear that this broad degree of regulation and responsiveness and sensing the environment and dictating our response was something unique that set us apart. But the other thing that also was a big surprise was that at an individual gene level, that there were a lot more variations in the gene than we expected. You know, most of us were taught way back in the day that there were dominant and recessive genes. You know, dominant being good, recessive being bad. If you had two recessives, it could be a really bad situation. And that was the belief, right? And it was kind of a digital situation, an on-off. You know, you either had a good functioning or a bad functioning gene, and it accounted for some of the diseases that were genetic and indeed related to one gene, where one gene issue could lead to significant challenges. For example, something like sickle cell, or something like Tay-Sachs, or something like cystic fibrosis. But the belief was going in that we would find a lot of other culprit genes that would explain certain illnesses. And what we have found instead is a lot of multifactorial illnesses, lots of gene things adding up on top of each other to create diseases. But in looking at individual genes, we saw these variants present, and some genes have 20, 30 different variants within them that we could call single nucleotide polymorphisms, or SNPs. Single meaning one little change, nucleotide meaning one of the compounds that's a nucleic acid, one of the four, and polymorphism is meaning many different forms. So, it's a little bit like thinking about the coding for a gene is a little bit like thinking about a written paragraph. But imagine, instead of our paragraphs, which have words of all different lengths, and we have 26 letters in our alphabet, and we have spaces, and we have punctuation, imagine instead that the code is written continuously, no gaps. All the words are 3-letter words, and they're spelt with only 4 letters apiece. So you can imagine that leads to a situation where translation of that is a little bit more difficult. So if you have a single misspelling, that's called a SNP. So you have a misspelling in your little 3-letter word. Now, some of those really have great consequences, and some of those really have smaller consequences. For example, if we're talking a 3-letter word like dig, D-I-G, and I have a slight misspelling and it's D-U-G, well, the meaning's pretty similar. If I say I dig a hole or I dug a hole, yeah, the context is there and they're pretty similar. However, if I change it to an O and I dog a hole, that starts becoming nonsensical. But it's still words, and it creates some sort of image in my head. Maybe I'm using my hands and doing this to dig the hole instead of using a shovel. You could see where you could at least form an image from it. And then you get to the point where DAG, D-A-G, or DEG, D-E-G, or D-Y-G makes no sense at all. And so it becomes nonsensical. So these little SNPs have the ability to change the amino acid that's called for, to change the meaning, which is really a translation to an amino acid. And different amino acids have different structures. Amino acids being the building blocks of a protein that's coded for, each amino acid's coded for by at least one unique one of these nucleic 3-letter codes that you get. And so what's interesting about this is that as you change which amino acid, you're going to get a change in structure, and structure begets function. So probably most of us have at some point played with like Thomas the Tank Engine trains with a child, or if we're older, maybe it was Lionel or Hot Wheels or something. You put together some sort of a track for a child at some point in life. And, you know, if you were reading the instructions quickly, or an older child was telling you which direction and they got it wrong, if you made a right-hand track turn instead of a left-hand track turn, then you're going to end up with clearly a different situation being present, and a track that may not work out. It won't link back at the end. Like if you turn right and do everything, well, you may end up with the end over here as opposed to over here, as opposed to here, where it's going to end up with a connection. And, you know, hence your child's not going to be able to run the train through an intricate circle if you get it wrong. But you can imagine that if you made a couple wrong turns, like if you made a right turn but then made an immediate left and made an immediate left, maybe it comes around that loop and it won't be so bad. The function may not be so bad. You know, a way to think about it a little bit is to think about perhaps, if you think about an enzyme and it has a pocket, and then it has an active site in the pocket where the work is done. So the molecule that it's working on comes into that pocket, and then the active site has some, you know, it either pulls two molecules together or it breaks a bond and shortens the molecule. And if you were to think about my arms representing the pocket, my head representing the active site, my arms represent a pretty good pocket. But if the message that codes down here that basically coded for a left-hand turn, if that message was wrong and it was straight, then the pocket's not going to be nearly as good. Things aren't going to fit in there. They're not going to stay as long, and the head can't be as good of an active site. Or imagine if it went the other way and came in like this, it may completely block anything from binding at all. So when you look at it, a small misspelling at one place can make a difference, but a couple misspellings can either make it worse or make it a little bit better. So back to the analogy, but imagine that the pocket is defined by my hands being like, let's say like this. We'll turn them outward. Hands like this, you know, and that's the pocket. And you can imagine that if something happened, one little change coming in like this could be really bad, but maybe two changes, if it came in like this, but I inverted my hand in a certain way, it would be okay. And two changes would maybe make up for what one change was. It's part of what makes it so difficult. You know, a gene that we're going to come back to talking about in a little while is the 5,10-methylene tetrahydrite folase reductate gene, which plays a role in a process called methylation. So, you know, that gene has 14 or more known variants on it, but only 2 of them do we really understand how much they affect us clinically in terms of the efficacy, the ability of that enzyme to have its outcome. So as we're looking at things, you know, this is a big source of variations and the one that we most frequently look at. Other sources are, you know, if you think about it, some of those misspellings could actually code for a 3-letter word, which would be the stop. So stop translating. So it's kind of like adding a period into a sentence early and not having anything after the period. You lose half of your paragraph if you stop at the period, or you lose half of your sentence if you have the period in the wrong place. So those sorts of changes, or changes in terms of what we call copy number variant, meaning that we carry a couple different copies of the gene, and depending how many extra copies we carry, is how much capability we have. You know, it's a little bit like back in the day when I was charged with doing pancake dinners at my daughter's school for the annual pancake dinner. If we had only had one griddle, we would have been really hung up in terms of making pancakes. But if we had 6 identical griddles and 6 sort of identical dads all doing a plus minus good job at making pancakes, we could go a lot faster. So similarly, that's the copy number variant, how many copies of the genes that you carry. And something interesting about copy number variant is that some of these genes actually have slightly different structures. It's almost like if we know that with this SNP, we have 70% effectiveness, and with this SNP, we have 100, with what we call kind of bookmarking, we could determine which one of those we're actually going to read. So let's get into that concept of bookmarking a little bit, and let's get into the responsiveness to the environment. So we know that there's all this source of variation that we didn't expect to see in the individual gene structure, which is going to have an outcome. So when that gene gets turned on, it's no longer digital, on-off, dominant, recessive. Now it's either wild type, which is the most common variant in the population, or one of the other variants that may be present. And it's almost analog. You know, you turn the dial on, you turn the dial off. You know, you get different volumes in between, like on your radio, as you turn the volume. It's the same with the variations in a gene. You may get 20%, 47%, 72%, 87% efficiency, depending which sort of misspelling that you have there and what variant of the gene you have there. And that's going to call for a lot of differences between all of us. It accounts for that biochemical individuality. But the other place where we get a lot of the variation is what we were talking about before in terms of the information that's put forward, the connection that's put forward, and the ability to, you know, respond to the environment. If I won the lottery tomorrow, or if you won the lottery tomorrow and we decided we needed a new house, we wouldn't just walk into an architect's office and say, I won the lottery. I want to spend $3 million on a house. Build me a house and I'll come back in 6 months or 12 months. You wouldn't do that. And the architect wouldn't do that. Instead, the architect would ask you a series of questions like, well, how many bedrooms do you need? How many living space? How much living space do you need? Do you like certain styles or not like certain styles of houses? You know, are you a Victorian fan or an arts and crafts or a Greek revival or a federalist or, you know, kind of a Southern California look, whatever it may be? You know, you get all these different options, and depending who you were, you would pick a different option for your house in these pieces, and yet you'd be still thinking about things. And chances are that the architect, particularly if they're building this sort of custom house for you, chances are the architect is going to give you a book to look at of the work that they've done, because you may not have settled yet on that specific architect. And that architect is going to go over and get this book out, and he's going to say, these are pictures of houses that I've bought. But because you hate, and frankly, I don't like Victorians, because you hate Victorian houses, well, I've put a binder clip on those pages, so don't even look there. But you told me you'd love to live in a Federalist home. Well, here's a couple right here, which I marked with a paperclip. What we do, because of our environmental responses, we can have short-term corrections of our DNA, which are similar to that binding and paper clipping. So we kind of determine in advance, in response to the environment, what's going to take place. an experiment that really demonstrated what this chemistry was Randy Jerdel's work looking at the Goody Mouse. So the agouti mouse is obviously a mouse, and it specifically has the agouti gene, which codes for coat color, and it's a yellow-furred mouse. And interestingly, these mice have been selected for their fur color, but these mice, in linkage with this fur color, are a series of gene mutations that that basically have led the mouse to be yellow-furred, overweight, to become diabetic, and to have a shortened life expectancy. And the question is this hard written into the genes? Is that mouse always going to express that? Or are there going to be processes that have an ability to change that? And what they did was they supported, and this is going back to the MTHFR gene that we were talking about in methylation. They gave these mice, and they were mice that they got pregnant. So the mouse was fed a special diet before and during pregnancy. They gave these mice a food plan that was rich in the things that support methylation. So there was choline, there was methionine, there was vitamin B12, B6, there was folic acid. They gave them a diet that would be supportive of good methylation, and they gave it to them while they were carrying their babies. And what they found was, unlike moms who had grown up with the standard kind of chow diet that's given in experimental settings and had grown up yellow-furred, soon to be obese, soon to be diabetic, and soon to be dead or dying sooner than their colleagues who weren't agouti mice. What they found was when they gave methylation support, the offspring were brown-furred, lean, didn't get diabetes, and had normal life expectancies. And interestingly enough, it wasn't that they had changed the gene, they had changed those binder clips and paper clips, those bookmarks on the gene, because if you then went and took that offspring and started feeding them the diet that mom had had before she got pregnant, within 4 generations, the mice were back to being the way that they were. So, the consequence of this is it's suddenly pointing out that we can take our genes and we can, due to environmental inputs, and food, for example, is this huge environmental input. Due to these environmental inputs, we can modify our genes. So, is there work that seems to support this in human beings? And indeed, this is an experiment which you're not going to do in humans, because you're not going to deliberately do something to change the bookmarks on a newborn infant. No institutional review board would allow you to do it. And also, you're not going to have an answer for years and years and years if you want to see final outcomes of the consequence of trying to change those bookmarks. So instead, what we have to do is we have to look observationally at the kind of the history of what's present. And if we do that, we can look back at the Dutch famine winter. The Dutch famine winter was the winter of 1944 into 1945, took place during World War II, took place while the Germans still occupied a good bit of the Netherlands, Belgium, and France at the tail end of World War II. And the American and British soldiers were advancing across France and getting ready to liberate Belgium and getting ready to liberate the Netherlands. And unfortunately, as winter hit, the Netherlands remained behind German lines. And the Germans were pulling back all the bounty from the agriculture and the harvests for those years to Germany to be used for the German people and for the military. And as a consequence, there was a famine. And the children who were in utero during their second trimester actually probably had their genes bookmarked, so that when they grew up with an increased risk of being obese, an increased risk of diabetes, an increased risk of heart disease compared to their peers, and maybe even an increased risk of schizophrenia. So it really was kind of this interesting change that took place, and it's not been watched for generations afterwards yet to see if the risk goes back down, but it was clearly demonstrated that that generation had different outcomes than their siblings. So children born before or after were the control group, and they had normal outcomes compared to the children who experienced the Dutch famine winter. It's really interesting, some of the early work identifying celiac sprue as being related to gluten actually came out of that, observing what took place during that time as well. A physician in the Netherlands actually noticed that his patients, who at the time were called celiac sprue, seemed to get better as the famine progressed. And that's partly because before the famine, they thought that they needed to feed children a diet that was rich in bland foods. And actually, they used a lot of white bread toast. So these kids continued to languish and struggle with this food that they thought was bland and easy for them to eat. And instead, when you took the gluten away and they had to eat other sources, they actually improved. And then when the famine was over, they worsened again as it became available once more to give them bread. So we have this situation in which we now recognize that there's a lot of variation. It comes from both individual gene variation and the interaction between the environment and our genes and what we end up expressing. And why this becomes really important is that with appropriate nutrition, support for methylation, support for various processes, that we can have different outcomes. To talk about just one of the genes that we can talk about in the office, we'll talk about the methylene, the 510 methylene tetrahydrofolate reductase gene, or what's frequently called MTHFR. It's just one step in the conversion of inactive folate, just plain folic acid, to the 5-methyl form of folic acid, which is necessary to convert homocysteine to methionine so we can make something called S-adenosyl methionine, which is a universal methyl group donor. So what is a methyl group? It's a 1-carbon atom with a couple hydrogens on it that can be added to all of those carbon chains, like an oil or sugar or a glycoprotein, which is partly sugar, partly protein. A lot of processes that take place in the body add a methyl group on. One of the places that's truly unique where we add methyl groups on, we add it onto DNA in order to bookmark it so that we actually close those pages so they can't be read at times we don't want them to. So, that's why this was so important when they did the Goody Mouse study, because they were methylating, they were bookmarking the DNA. Well, it's also really important because it plays a lot of roles in basic chemistry in the body. If methylation isn't happening well, then we know homocysteine levels, another amino acid, levels are actually high. And high homocysteines have been associated with high blood pressure and heart disease and neuroinflammatory disease and risk for stroke. So clearly we don't want to walk around with high homocysteine levels. And so, hence understanding this gene-gene interaction plays or gene nutrient interaction plays an important role in us being well. If the gene doesn't work as well, so the two SNPs that we know the most about are C677T, where a cytosine is replaced by a thymidine. In this, or maybe it's cytosine replacing the thymidine, whichever. When you think about it, if you have one copy of that, you have a slight reduction. If you have two copies of that, you can have as much of that gene with that SNP. You can have as much as a 60% reduction. And if you have one copy of the C677T and one copy of the A1298C, you're going to have about a 40% reduction. So, as you start looking at this, you can learn something about how well things work. And considering methylation plays an important role in estrogen metabolism, in neurotransmitter metabolism, in the modulation of oxidative stress, and even in terms of endothelial function, how well our blood vessels dilate, it becomes crucially important to know the methylation status when dealing with certain conditions and knowing how to modify it from a nutritional perspective. The other place that, this kind of genetic variation starts playing a role, and what we really have to remember, what this teaches us, is that even genes that may be maladaptive in a certain setting, in a different setting, may actually be okay or actually be good for us. On the chromosome, and we label chromosomes with a combination of the number and then a letter and then another number, which defines the place on that chromosome or the region on the chromosome. On chromosome number 9 at P21, there's some single nucleotide polymorphisms, which we don't really know what they do, but they code for an increased risk for heart disease. Interestingly enough, if you have those abnormal variants, if you have the SNP of that gene and you eat the typical Western diet, you are going to have an increased risk for heart disease, more so than if you didn't have a copy of that gene. But if you ate a plant-based diet, a rich diet, then it ends up being a situation in which you actually offset that increased risk. Eating multiple servings a day of fruits and vegetables, like greater than 4 or 5, restores your risk to the baseline. So, we know important differences and the such, like methylation SNPs may play a role in how well we tolerate certain preservatives that are used, for example, in vaccines. Thimerosal, which interferes with methionine synthetase, has been looked at as a culprit by some in kind of affecting methylation and looking at autism, where children have an increased likelihood of carrying SNPs which show methylation abnormalities. When they take thimerosal, there seem to be at more of a risk of having impaired methylation, which may have something to do with the risk for autism. But, as we start looking at the vaccine, for example, for COVID, people are asking me about it now, like, should I get it? Should I not? And I'm saying a lot's going to be determined by what kind of vaccine it is. Is it, you know, an RNA or DNA sourced vaccine? Is it a live vaccine? Is it an attenuated live vaccine? Is it a killed vaccine? So all those things will play a role, but so will play a role in terms of the SNPs that are present. Because if you have issues and they use thimerosal as a preservative, then we're not going to be so excited perhaps about that as a vaccine for you. And then the other important utilities that we're already, you know, identifying, you know, we know that when it comes to drug metabolism, that there are certain what we call cytochrome P450s, certain enzymes that metabolize, but we know that our genes that code for these can code for greater or lesser amounts or greater or lesser activity. So we have uniquenesses when it comes to how much we can tolerate. If I, you know, like, for example, caffeine, not a drug, but something that we take in all the time, and it has active effects on our physiology. If I'm a fast metabolizer, I can drink caffeine all day long and not notice it. If I'm a slow metabolizer, 1 cup in the morning may actually impair my sleep in the evening. So you can imagine that depending on that sort of activity, or depending on other things that we may take or the interactions, knowing what our SNPs say about our ability to metabolize will be an important place for us to visit. And we're able to do that with genes at the moment. You know, we don't really have the software programs that completely have integrated it, but there are places that we can look and say, oh, this drug is metabolized by that SNP or that cytochrome. And if you have that SNP of that cytochrome, you're going to be less effective. So you think about changing the dosing. And similarly, you know, we're getting to the point where we're probably going to be able to predict diets down the road. You know, like, you know, here we have a specific diet, like one we like to use is the high protein, high phytonutrient diet. And if that's indeed the case, then, you know, who's going to benefit from that? So if we could get some genetic information right up front, and you filled out a questionnaire and we know something about your SNPs, then, you know, we may be in a place where, you know, we know that a low-fat diet would be better for you than a low-carbohydrate diet. And we'd make the appropriate choice to kind of help you on the path to wellness. So appreciate you all listening to me. If there's any questions, you could type them quickly. I just want to point out 2 upcoming, well, first of all, we'll be back in a month. And the topic next month is going to be healthy aging in the modern world. Second thing, tonight I'm speaking for Harmony Hill Cancer Retreat Center and giving a talk entitled Uncovering Certainty and Rediscovering Soul. I hope you'll join me there. It is a fundraising event for Harmony Hill, but they're a very deserving organization. And finally, Michael Stone and I on our Monday afternoon coffee breaks, 4 p.m. Pacific Daylight Time, starting this coming Monday, are going to do a 3-part series on compassion and hate. So I hope you've had a great day. Hope you're heading into a good weekend. Be safe from the smoke, and we'll see you next time. Thank you.